Now, as they say, for something completely different. So we're a pure stem cell therapeutic development company. That's all we do. So we're a biotechnology business quoted on AIM. We've actually been around for around 20 years, which tells you something about the length of time it takes to develop advanced therapies. And I'll get into some of the reasons for that shortly. But we are a leader in our field, which is good. And primarily we're developing what's known as allogeneic stem cell therapies. So these are cell therapies that don't depend on the patient's own stem cells. Essentially what we're trying to do is produce a standardized biotherapeutic which can be administered to any patient that rocks up with the diseases that we're looking to treat. So it's non-patient specific or off the shelf. And in our field that's going to mean lower cost of goods and a greater chance of delivering something that's going to be of interest ultimately to a commercial third party should they choose to acquire these technologies in due course. So there's a real commercial imperative behind what we do. It's harnessing stem cell science and trying to make ultimately money for it, as well as treating patients that currently have no alternative treatments available for the diseases we're targeting. We're also at the forefront of something called exosome-based nanomedicine. I'm not even going to get into that today, partly because I'm not the scientist around here, and it's also very early stage. So I'm going to focus on two of our most advanced programs today. A treatment that we're developing for stroke disability, where we've recently announced data, and also another treatment that we're targeting against retinal degenerative diseases. So those are the two I'll focus on today. We have two platforms, and you can see how we're exploiting those two platforms. One's called CTX, and the other one's called human retinal progenitor cells. And every biotech has to have a pipeline chart, so here's ours. And you can see that we're taking these programs through a clinical development pathway. Phase 1, Phase 2, Phase 3, and then hopefully all being well through to market. There's a number of ways you can do that, and in advanced therapies the clinical development pathways tend to vary a little bit as compared to more conventional small molecule drugs or large molecule biologics. But nonetheless, that's basically what we're trying to do, garner what we hope will be well-controlled good data, clinical data from well-controlled, well-designed clinical trials. If those data are positive, that will build value in the business. That's how the biotech model works. Biotechs typically, especially on this side of the Atlantic, tend to get acquired or they license their programs at some point through that clinical development process, or occasionally they actually go forward and launch these products. And actually that happens more in the US, where companies tend to be better funded and they can take their products all the way through. Clearly, if they can do that, there's more money for shareholders at the end of the day if you can actually get that far. So why stem cells? And this is the quick science lesson. It's the only bit of science in the presentation. Stem cells are nature's repairing cells. We all have them. They're associated with restoration of function after injury or after disease. And there's been a lot of development work to try and harness the power of stem cells and try and convert them into something that's therapeutically beneficial. And the big issue is, how do you actually scale stem cells? How do you turn them into ultimately a therapeutic product? And there are a number of players in the field that have been able to make really good progress in that direction, and we're one of them. So what we're trying to do here essentially is to offer a cell-based treatment that has the ability to treat and possibly even reverse some of the symptoms and indeed the underlying causes of diseases that more conventional treatments just cannot get at. And that's the great promise of stem cell therapy. And with that, I'll move straight on to the more businessy aspects. As a company, we're pretty well backed and funded at the moment. Woodford Investment are the largest shareholder at the current time, as you see here. And if you're a small biotech company not making revenues but actually raising money and then spending it on those clinical development programs, it's important that you have a war chest, if you like, to be able to do that so that you don't sort of run out of cash at the critical moment. And in that regard, we're pretty well placed. At the last reporting date, which were the interims on 30th September last year, we had just over £60 million in the bank. And that will fund those therapeutic programs over the next couple of years at least, to points which we would regard as being meaningful clinical development milestones, which we hope, if they're positive, will build real value in the business. And those key investors understand that. They are very well attuned to that model within the field of life sciences and biotech in particular. So that's where we are sort of financially. I'm happy to answer questions on some of the more sort of detailed aspects of the finances, our burn rate and all that kind of stuff, if you wish, at the end. But moving swiftly on to the programs in a second. Before I do that, just a quick nod to the senior management team that we now have at ReNeuron and our non-executive board. And we essentially now have all the core functions that we need in-house. We've invested quite heavily in our staff. And also our non-executive team is, as far as we're concerned, second to none in terms of what they bring to the business. They are all highly seasoned entrepreneurs with many, many years combined experience in life sciences and biotech development in particular. So just moving on to CTX, which is I suppose our core cell-related asset. What is it? Well, CTX is a GMP, good manufacturing practice validated, cryopreserved human stem cell line. It was derived over a decade ago and expanded by way of genetic modification. Essentially, it's a vial of stem cells. So a treatment of CTX involves administration of several million stem cells directly implanted. Those cells can be cryopreserved, stuck in a cryoshipper that you see in the bottom left there, and shipped and stored at the hospital site to be used when required, currently under a 6-month shelf life. So what we're trying to do here is really convert stem cells, a cell-based product, into as close as we can get it to a more conventional biotherapeutic paradigm, like biologic drugs, for instance. It just so happens that it's essentially a living cell product when thawed. And so the hospital centre and most large hospitals have pharmacies that can work with these types of therapies. They merely thaw, maybe dilute if necessary, and then administer the patient when required. So that's essentially the essence of CTX, what it is in terms of a cell-based approach. So now moving on to the programs, and I want to focus quite a bit on stroke disability, which is our most advanced program. And what we're trying to do here is address the consequences of stroke for those that survive their stroke but are left disabled. And if you get a stroke, you've got a 1/3 chance of dying from it, not surviving. And if you survive it, about a 50-50 chance of being left with a residual disability. And what we're looking for CTX to do is to try and mediate the effects of that disability, to reduce the patient's reliance on health and social care, to make them less dependent and to lead a more independent quality of life. And there is clearly a very strong patient imperative to that. And there's also a very, very strong pharmacoeconomic argument for that as well, driven by the huge health and social care cost burden that disabled stroke patients represent in the developed world. So just moving on to where we've got to with this program, and again, going back to that clinical development process, phase 1, phase 2, and so on, we've already run and completed a phase 1 study here in the UK in 11 disabled stroke patients. The results actually were published in The Lancet last year. It was primarily a safety study to make sure that these cells were safe when implanted. And we saw no cell-related or immunological adverse events in these patients. And encouragingly, we did see some evidence of efficacy against these patients' stable baseline level of disability. So it was what's known as a single-arm study where we're monitoring those patients against their stable baseline disability pre-treatment and then taken 2 years out post-treatment. So much for phase 1. We recently announced data actually last December, a couple of months ago, with our next study, our phase 2 study, which we called PISCES II. The first study was called PISCES I, imaginatively. That's an acronym, but I won't even tell you what it is. The PISCES II study was really focusing more on efficacy of the treatment, as phase 2 studies typically do, to see in a larger group of patients whether we could confirm an efficacy signal in disabled stroke patients. So in this case, it was 21 patients, male and female, quite a broad age range, as you see here, and treated between 2 and 12 months after their stroke. And all of these patients were characterized by having very, very limited upper arm function. I don't know if any of you watched the Andrew Marr programme on BBC Two last night. I think there should be a link to it on our website now. But it did feature a patient from this very study, and indeed an interview with the principal investigator, Professor Keith Muir, who ran the study based at Glasgow Southern General. Essentially what we're trying to do is measure patients' ability to perform useful tasks and to measure them against various other measures of dependence and activities of daily living and so on. And I'll come to those measures in a second. Again, what we're doing here is implanting 20 million CTX cells directly into the brain on the affected side, and then monitoring those patients post-treatment for 12 months. So in terms of those measures I mentioned, and again, this is quite a busy slide, and I don't want to go into these in great detail. What I'm going to do when I show the data in a second is just focus on the top two. The top two, modified Rankin Scale, or MRS, and Bartel Index, BI. And those are important to us because these are respectively measures of dependence in these patients and also measures of their ability to perform activities of daily living. And those two measures are important because from a regulatory and a clinical perspective, they are typically used as primary endpoints in later stage clinical development, which is where we're about to go into. So getting a hit on those measures is actually pretty important because it's very likely that we'll be using a combination of those measures in our next study, our phase 3 study, which we're hoping to start later this year. We also used a couple of other tests as well, ARAT and Fugel-Meyer, so a pretty full suite of efficacy measures to test these patients on pre and post treatment. In terms of the status of this study, where are we currently at? The patients are actually treated, the median treatment time post-stroke was actually around 7 months, but as I said, the range was actually between 2 and 13. So sometime after their stroke, these patients were treated, so they've stabilised in terms of their disability, but have that residual stable deficit thereafter, in much the same way that Andrew Marr had, if you saw that programme last night with his affected arm and leg. 8 UK sites were involved in this study, and in terms of follow-up so far, all 21 patients have been followed up for 3 months, around 10 for 6 months, and 3 patients are actually all the way through their 12-month follow-up when we announced the data in December last year. So what did we see? Well, on that first scale, modified Rankin, which is a measure of general dependence, it's a fairly tight scale ranging between 0 and 6. The lower the score, the better in the case of this particular scale. And what we were able to do is out of 21 patients, we were able to move 7 of those patients down at least one measure, one notch on that scale. And any 1 point movement on that scale is clinically significant within the context of clinical development studies like this. So that response rate, 7 out of 21, is very, very interesting to us. That's a level of response that makes this program interesting from the point of view of further clinical development. It's high enough to warrant further investigation. So we were very, very pleased to see this. And indeed, we actually managed to get 1 patient down by 2 categories, which was very surprising, actually, and encouraging. And this is just a different way of showing the same data, really. It's a shift analysis in colour. So as you can see, what we're hoping to see here over time is a gradual shift away from the red and the green and more blue and purple coming onto the graphs over time, with obviously the blue and purples being a lesser level of disability and the red and the blue being more disability or a higher level of dependence, if you will. So far so good. And the other thing that's worth noting is we haven't seen any patient so far revert on any of these treatments throughout any of the follow-up period. So if we manage to keep to that, we might expect these data as they mature to actually show a better level of response over time as well, because of course we still have quite a number of patients to go right the way through to their 12-month follow-up point. And we'll obviously keep the market informed as we garner more data from this study. Similarly on Bartel is a measure used to monitor patients' ability to perform activities of daily living. Can you get from bed to chair, from chair to toilet, and so on and so forth. And here again, a similar level of response, indeed slightly better actually on this score. because rather, again, without getting into the detail too much here, of the 21 patients, 6 of those patients already had a Bartel score that was high enough such that they could not actually achieve a level of change that was going to be clinically significant anyway. They just weren't disabled enough, if you will, on this measure. So that left essentially 15 patients that could be evaluated on this measure. And again, we saw a clinically significant change in 8 of those 15 patients. So again, a really good response rate in studies of this nature, and something that gives us confidence to move this program forward. In the interest of time, I'm not going to go through the Arat and Fugel-Meyer scales. I'm happy to talk more about those afterwards if anyone's particularly interested. So what were the conclusions for us as a business in terms of this study so far? Well, we were surprised and positively surprised at what we saw in terms of responses, especially against those key measures of dependence and activities of daily living, and unmodified Rankin in particular, and that's probably quite likely to be the primary endpoint that we'll go on to use in a phase 3 study, as other developers have done in similar phase 3 studies when looking at disabled patients, not necessarily in stroke, but in general in other trials of disability, or studies of disability, I should say. Also, from a safety perspective, again, treatment was well tolerated, no adverse events attributable to the cells. There was one death actually in this study so far due to sepsis a number of months after treatment, but that was assessed as not being attributable to the treatment. So that's now led us, as I mentioned earlier, to work through the design of a phase 3 study, a pivotal study, something that will really confirm everything and get us to the point where, all being well, we can think about getting this registered for market approval. And the aim here really is to, again, focus on patients with established disability, but this time this study will be a randomized control study. It will involve placebo surgery. Some of the patients will get the cells and some of them will get the procedure, but they won't actually have cells injected into the lesion, or close to the lesion rather. And we have to do that, but that is something that we did not want to have to contend with until we had a decent phase 2 efficacy signal, which we now have. And the regulator, again, makes the discussions with regulators a lot more straightforward if you can offer them decent data that then warrants going into a placebo-controlled study, which we will have to do to get this right the way through its clinical development program. Primary endpoint, as I mentioned, will be measures of dependence and activities of daily living, so Rankin, Bartel, and the like. And those are, as I also mentioned, are measures that are known to physicians and regulatory authorities around the world. It's likely to be predominantly US, but perhaps with some European sites, this phase 3 study, looking to start it at some point this year, as I mentioned, and all being well, data readout sometime in 2019, probably towards the end of calendar 2019. Okay, so that's stroke. So in the time I've got remaining, which isn't much, just a quick word about the second platform, our human retinal progenitor cells. So a completely different asset that helps to mitigate risk against the CTX programs. And this is based on our HRPC retinal cells. Here we're looking for those retinal stem cells to essentially either rescue existing photoreceptors, the rods and cones in the back of the eye that give us vision, and also potentially to replace lost photoreceptors, so to differentiate into those photoreceptors that have been lost in these retinal degenerative diseases to restore a degree of vision in these patients. We've been working with a lot of very clever collaborators over the years to optimize this program preclinically to the point where we've now got a first clinical trial running in the US. The target for that trial is retinitis pigmentosa. RP is an inherited degenerative disease of the eye involving loss of peripheral vision initially, as you see in the image there, and ultimately leading to blindness. And this is an orphan disease. It has orphan drug designation here in Europe and in the US. And what that means is that it's a fairly limited patient population, actually quite a large one in this case, as an orphan, 275,000 patients in the US and Europe combined. But orphan status diseases are favoured by the pharma industry now. Why? Because they are quicker to get to market, because the patient populations are smaller and the need is greater typically in those patients. the regulators have made concessions so that you can move orphan status products through clinical development more quickly. And this is what we hope will be the case for our program in RP. We have a phase 1-2 study ongoing in the US at the moment, and that will be followed by a phase 2-3 study, which we'd hope to start next year, and hopefully that will also read out in 2019 calendar as well. And that leads on to the milestone chart. So this is purely a chart that shows when will we expect to get clinical data readouts of significance over the next 3 years that we regard as being meaningful clinical milestones that are going to make the programs have value, to be attractive to potential licensees or even acquirers, or give us the ability to take these programs right the way through to market ourselves, should we so wish. And these are the milestones that we've identified across the programs, including 1 or 2 that I haven't actually spoken about today. So just to wrap up in the minute I have remaining, we are a differentiated stem cell technology business. We've cracked a lot of the technical issues of how you go about commercializing and scaling stem cells into therapeutic products. We have a strong competitive position because of that. Very high barriers to entry, as I'm sure you can imagine. We're targeting very significant diseases, either because they are very large patient populations or they have significant unmet need. And we're developing a high-value clinical portfolio. Our programs are at clinical stage. We're well-backed and well-funded, which we have to be able to run those very expensive clinical trials. And we have a great leadership team now as well to get the job done. And those milestones, if they're positive, any one of them actually should, if the data is positive, bring real value to the business, much in excess of the current value that the business carries on the AIM market. And with that, I'll take some questions. Thank you very much.
Any questions?
Evening. Can I ask you what your end game is? Do you intend to develop the business, and if so, what's your vision? Or do you intend to sell it to one of the big boys, and at what point do you think you would be in a position to do that?
The best way to get a deal is to run your business as if you intend to launch those products. And indeed, there's ultimately no reason why we shouldn't be able to do that. These are specialised hospital products that won't require enormous sales forces that you might imagine would be required to launch, a pharmaceutical product on a global basis. We don't necessarily need that, and indeed our CEO has launched pharmaceutical products in the past. That's why he was hired a couple of years ago to bring those skill sets into the business. What will actually happen may be something completely different, and clearly our shareholders will determine that to a degree. If we're made an offer either through a license arrangement or a full-scale acquisition, that is something clearly that if the terms are right, we would need to put to our shareholders. But the best way of getting to that and building value is to run your business on the basis of launching those products, and that is exactly what we're doing.
Gentleman in front. Can you just wait for the microphone just 1 second? Thank you.
How many centres are going to participate in the phase 3 trial in Europe and US?
TBD, but we reckon it will be at least 30. So generally, I mean, the way to run it is if you sort of divide by 7 in terms of a per patient per month recruitment rate. And so depending on how long you think you've got to actually recruit and then follow up the patients, you can kind of work back and determine how many sites you actually need. And then you have to consider the territory you're working in and how many centres can do this stuff, or at least do it within the context of a clinical trial. But we reckon probably around 30, maybe let's say between 30 and 40.
And the next question is, what's the potential for the exosome to generate some revenues for ReNeuron in the near future?
That's a good question. Fair to say that we are still evaluating how best to exploit that platform. There's a plethora of opportunities there, and the one thing we do know is we can't do it all ourselves. We're not funded to do it all ourselves. It would be a distraction, and for that reason, maybe there are some opportunities to collaborate and work with others. I can't really say much more than that because I don't really know at this stage.
Gentleman at the back.
Hello, Michael. The question I've got is to do really with the manufacturing plant as such. As I understand it, we have a set of stem cells which presumably to keep to a standard you have to keep reproducing, like a culture, like brewers do with yeast. What sort of manufacturing facilities do we have, and is it licensed out in any way?
So currently what we have, and this will probably be the case until we launch or we are launch ready, we have a reliance on contract manufacturers around the world. So there is a fairly limited pool of contract manufacturing organisations that know how to do this stuff, and essentially you transfer your technology into them. So we're currently working with 3, 2 in Europe and 1 in the US, and that we may actually expand that a little bit as well. But those organisations are expert in terms of, you know, essentially manufacturing biologic-based products, including cell-based products. So they have the technology to actually scale the cells, to culture them to the quantities required, to the quality standards required, and to the using the technology that we have transferred into them to enable them to do so. Ultimately, once we get to launch or close to launch, we want to bring that in-house, at least for the CTX product. And that is why we moved the business to Wales a couple of years ago, or a year ago I should say, in order to build out a manufacturing site there to GMP and to invest fully and bring that into our own hands. Why? Because ultimately if we do want to, let's say we do want to sell the business in due course, we are made an offer, it's going to be much more valuable if we can offer the means of production as well. Because large pharma wouldn't know how to do this stuff. You know, they're marketing machines and they do have research capability and so on, but they know what they know and they know what they don't know. And it's probably the case that they won't know and won't want to have to learn how to do this stuff at a manufacturing level. So we'd like to be in a position to provide that to them, and our Welsh site, you know, would be able to fit into the portfolio of a pharmaceutical company's infrastructure if, you know, if we went down that route. Okay. And it also offers great cost advantages if we can bring it in-house as well. CMOs are very expensive.
Thank you very much.
Any other questions, anybody? Gentleman there. The final question, by the way.
Yeah. Hi, Mick. I'd just like to ask if you have enough money with the cash burn to take the CTX right through to the end of phase 3?
Probably not, but we won't need much to get there. And the investor base that we have at the moment is more than capable of following its money. Those lead investors, again, I can't speak for them, but my sense is that they have invested in the story to see the story play out. So we probably will fall a little bit short, but not by much. And I don't think we'd have too many issues in topping up to the degree that we needed to get both of the programs right the way through phase 3. And I'm thinking here about not just one, but the 2 core programs, because they're going to kind of run somewhat alongside each other. And it's important that they're both adequately funded.
Thank you.
Thank you very much. Thank you for all the questions, and thank you very much indeed, Mike.